time. So I hear myself in my ear after an ever so slight delay. Right. I need to share the link for this event. Oops. I guess I'm going here and doing this. All right, so let's go to here. Let's click on this. Copy this link. And then go to, oh, it's going to be messed up if I do that. Stop. Shoot. Okay, close that. Okay. Here's my login. Plus. Oh, what the hell? Remove. Thought for sure. Oh, well. This gives y'all an email, I think, doesn't it? External URL. Paste. 10:15. Add item. And publish. There goes your announcement. What? No. Which probably means you get 2 notifications. All right, that is done. So today I am going to go through Operon theory, and I need to reduce the volume of this. Okay. I think that'll work. Is that a background hum? Let me check. Okay, that was the fan in the bedroom, I guess. Nope, I don't hear it now. Okay, great. So, yeah, this morning I'm going to go through Operon theory, which is a type of gene regulation used by bacteria. And there are a couple of examples in here that you will likely have to repeat 1 or more times. Excuse me. In different settings, and it might be something they ask about on the MCAT still. I don't know. It's been a long time since I took that. Okay. So the learning objectives here are to compare inducible operons and repressible operons and describe why regulation of operons is important. Now, the second one you can probably already intuit on your own without me saying anything. Any kind of gene regulation is important so that the organism hosting those genes is able to respond to changes in their environmental circumstances. and you don't want to burn resources just creating enzymes you don't need all day. Anything that did that didn't persist. In fact, if we give E. coli one of those conjugative plasmids, one of those fertility plasmids that have ampicillin resistance on it, within 100 or so generations, the E. coli will develop their own switch for that, so that they're not just producing the beta lactamase all the time, because it doesn't have to be always on. It just needs to be on when you're exposed to the antibiotic. But yeah, 100 generations, I think it was, on average. Okay, an operon. So in prokaryotes, structural genes of related function are often organized together on the genome and transcribed as a single unit under the control of a single promoter. The regulatory region of the operon includes the promoter, and here we go, the promoter and the operator. In some cases, you have a repressor that will bind to the operator, and in which case the entire operon is turned off, and your regulatory genes will be somewhere else. Now, the repressor, alternatively, you can have an activator that binds to the regulatory region that will enhance transcription. So in general, to recap here, we're talking about a single message that contains multiple genes. That's polycistronic, if you remember from last time. Okay, transcription regulatory molecules. This is our cast of characters that are involved in these activities. But we have transcription factors, which are a huge factor in eukaryotic transcription. And these are simply proteins encoded by regulatory genes that influence transcription. Of transcription factors, in general, we can have a repressor, which is a transcription factor that suppresses transcription, and it generally in response to external stimulus, and it works by binding the operator. And then the activator, another type of transcription factor, and it increases transcription, generally by facilitating the binding of RNA polymerase. Then we can also have an inducer, and that's a molecule that activates or represses transcription via interaction with the activator or repressor. And that'll become more clear momentarily. Oops, over here. There we go. Wait, that's right. Operon classes. So any operon that is being continuously expressed, that is unregulated, is called constitutive. Right here, constitutive operon. Now, if instead it is regulated and is not continuously expressed, it is inducible. We have 4 kinds of inducibility. So again, this is controlled by repressors, activators, and via their interaction with inducers. So if an operon is negative inducible, then the repressor is normally bound to the operator. So the default condition is repressed. And this is true of the lactose operon. Well, that codes for processing lactose, the sugar lactose. So that is negative inducible. The default state is repressor bound to operator. Now, if it is a negative repressible operon, the repressor only binds operator with a co-repressor, and this is true for tryptophan. If the operon is a positive inducible operon, the activator is normally unbound, requires the inducer to activate. And again, this is generally through the recruitment of RNA polymerase. If the operon is a positive repressible, then the activator is normally bound. The inhibitor will force release. And the examples we're going to be going through are going to be for the lac operon and the trip operon. Okay, the lac operon, this is an example of a negative inducible operon. So the lac operon, we have lac Z, Y, and A. These are the three structural genes that are needed to degrade lactose. And they're all next to each other, operating under this promoter and operator. When there is no lactose present, the lac repressor will bind the operator, and none of these genes will be transcribed. When lactose is present, the actual it's allolactose that will bind to the repressor. The repressor's conformation will change. It will be released from the operator. And this will allow RNA polymerase to bind and start transcription. So again, this is simply lactose present, a repression is relieved, RNA polymerase can bind, and it will be at a low rate, slow rate in this case. All right, so here we go. So the other aspect of the lac operon, the regulation thereof, is the interaction with cyclic AMP. So when glucose is less available, ATP will be reduced, and that causes CAMP levels to be changed, and that signals glucose depletion. So this is the backstory for the other part of the regulation, the interaction with glucose concentration. Okay, so we have the other aspect of lac operon regulation is catabolite repression. And this involves the interaction with the CAP protein. And this is the catabolite activator protein. Did I have that up there? No, I don't. I'll be writing it on the board. Okay. But CAP. So without cyclic AMP, the catabolite activator protein does not bind the promoter. So transcription, provided lactose is present, is occurring at a low rate. When we have cyclic AMP, and this is because glucose is low, the CAP will interact with the CAMP and then bind the promoter for the lac operon. So the CAMP-CAP complex will stimulate the RNA polymerase and increase the synthesis rate. So if the repressor, so if there's still no lactose, then obviously the repressor is still there. So it doesn't matter if there's the CAMP-CAP complex bound to the promoter. It simply won't be able to proceed. So this is the aspect of the lac operon that is positive inducible. Okay, glucose repression. Do you remember why I included this? Oh, right. So we have ATP interacting with adenyl cyclase. If you are in cell or going to be in cell, you're going to talk about adenyl cyclase and ATP and CAMP. This was probably mentioned or may have been mentioned for you in 115, talking about the cell cycle, but it may not have been. Okay, so anyway, if there's a high glucose level outside the cell, then inside the cell, the adenyl cyclase will be inactive. So we have low cyclic AMP, so largely CAP will not bind to the promoter of LAC. If there's low glucose, we have active adenyl cyclase, so some of that remaining ATP is getting converted. Well, we have high CAMP, we have high cyclic AMP, and then that will activate to increase the transcription rate of the LAC operon. All right, so here's a quick little graph, throwing this together, a little table. So if we have glucose, CAP is not binding. That is true in any condition. If we don't have glucose, then CAP will bind to the promoter of the LAC operon. If we don't have lactose in the presence of high glucose, well, okay, so this part doesn't care about that part, but here's just the result of these conditions. If we don't have lactose, the repressor is bound to the operator, so there's no transcription. If we have glucose and we have lactose available, there will be some level of transcription. It'll be like basal. If we have low glucose, no lactose, no transcription. If we have no glucose, yes lactose, then we will have transcription at the full speed rate. Okay, so here's what it looks like on the E. coli growth curve when it has to switch from glucose to lactose. So this is the result of being grown with both. The glucose gets depleted right about here, and then it takes a little while here to readjust to start using the lactose. Note how less steep the growth curve here, the exponential portion is for lactose. So obviously lactose is less preferred, and the growth rate here appears obviously higher. All right, so that's the basic LAC operon, negative inducible with catabolite repression. So TRIP represents a repressible operon. So when there's no tryptophan, the TRIP repressor is dissociated from the operator, and we have synthesis. When tryptophan is present, tryptophan will bind the repressor. Tryptophan repressor binds the operator, and the RNA synthesis is blocked. So this requires tryptophan to be an active repressor. So these, so these 5 structural genes are located next to each other. Let's see what tryptophan repressor proteins. Yeah, okay, that's what I just said. Okay, so negative control of the TRIP operon. So with no tryptophan, no TRIP repressor exists. It's just the inactive aporepressor. So this is a little more molecular detail on that operon, on that process right here. So if the aporepressor binds tryptophan, changes conformation to gain high affinity to the TRIP operon, that's down here. So tryptophan is a co-repressor of its own making. Is that all we're talking about there? Now, that might be. There's more detail here in the ribose switch, which I think I talk about later. Yes, I do. Okay. So in the global responses of prokaryotes to harmful or potentially harmful conditions, we have a group of chemicals somebody decided to call alarmones. I can't decide if that's cute or not, to be honest. I don't know. I think maybe it's useful. So these are small intracellular nucleotide derivatives that are able to change gene expression, stimulate stress response. And then I mentioned one of the alternative sigma factors. Here are some others. We have, okay, so 54 was starvation, which is my favorite. We have 19. So iron is another big issue for a lot of prokaryotes, particularly in places like the ocean. But iron isn't freely available in a lot of environments, so there's a lot of metabolism around iron starvation as well. So sigma 24 is around heat stress, sigma 32 around heat shock, and then sigma 38 is involved in stationary phase starvation response. So here's a little bit about ribose switches. These are really pretty cool. So prokaryotic messenger will have these features, and they combine to small intracellular molecules, stabilizing certain structures, influencing either the completion of the synthesis of the messenger, which is pictured on the left, or the protein made using that message. So in this cartoon, it is on. We have this anti-terminator stem loop, so there is no interference. So this part of the message right here is available. Then you can have a small molecule. In this case, it's acting like a repressor that interacts with the ribose switch region here, causing the anti-terminator stem loop to be lost, basically, because this whole area reconfigures. And then the terminator stem loop is able to form, preventing transcription from happening. So that's on in transcription. In translation, we can do something similar. It's the same thing. It's just you're blocking the start codon from having access to the ribosome, or vice versa. But yeah, so these are ribose switches. And this brings us to the other aspect of the regulation of the tryptophan operon. There we go. So what are we looking at here? So when tryptophan is plentiful, transcription of the short leader peptide encoded by TRPL proceeds. Where's my TRPL? Here it is, TRPL. It will proceed the terminator loop between regions 3 and 4. Here it is. It forms and transcription terminates. When tryptophan levels are depleted, translation of the short leader peptide stalls at region 1. Here we go. We stall here. And allowing regions 2 and 3 to form the anti-terminator loop, and the RNA polymerase can transcribe. So if we don't have tryptophan available, the ribosome will be stalled for long enough for the anti-terminator stem loop to form, allowing translation to proceed. I think that's pretty cool. That's called attenuation. That's where secondary stem loop structures form within the 5' end of a messenger being transcribed. Determinative transcription will complete the synthesis of this message, and if the message will be used for translation. That's attenuation. So this is attenuation by presence or absence of the tryptophan. Well, the short leader peptide, ultimately, directly. All right. So next, I think I mentioned these. I might be mentioning them again. This is more in eukaryotes. So upstream activator sequences or enhancers. We, as eukaryotes, use these frequently. You'll have this enhancer region. See here with the cartoon here with the little distal control elements, and this is hundreds to thousands of nucleotides upstream of our gene. And of course you'll have a DNA bending protein involved too, and you'll be able to make this. What else here? So two different genes may have the same promoter but different distal control elements, enabling differential gene expression. Oh, I hope you're not hearing that annoying optimize your PC bullshit from Dell. Close. Okay. Clinical focus resolution. Okay, Mark survived his bout with necrotizing fasciitis. He has to undergo skin grafting followed by physical therapy. His leg will not likely return to full strength. Laboratory testing revealed that the causative agent of Mark's infection was a strain of the group A strep. So it was reported to the state and the CDC. A strep isolated from Mark was analyzed more thoroughly for methicillin resistance. Methicillin resistance is generally encoded, so encoded by a gene, like a specific gene product, instead of being a mutation making the the pathogen more resistant. It's an actual specific gene product, like beta-lactamase is for ampicillin resistance. So let's see. What do we care? Do we care about this? Let's see. Blood flow is restricted. No bleeding. So they used intravenous antibiotics and probably had to debride the crap out of it. They discovered some virulence genes shown to be encoded on prophages, indicating that transduction was important in this particular case. So Mark did survive this. He didn't succumb. But I guess you could justify intravenous antibiotics in this case because you're trying to manage potential sepsis. Okay, that is the end of chapter 11. I'm probably going to go into chapter 12, so I'm not here. Let me minimize this. Modern applications of microbial excuse me. Modern applications of microbial genetics. Control of growth. I want to get us into a bunch of these as possible, so we might skip one or two of these chapters. But it's hard to choose though, because these chapters are a lot of fun. Let's take a quick look at modern applications of microbial genetics. I think this is kind of important because it talks about a bunch of the technology involved. 100 freaking slides. What the freak was I thinking? Oh yeah. Wait. This would be more fun in person. I'm gonna mention these things because what I did not like, it was very embarrassing and uncomfortable a little bit, or at least awkward, is that when you go into a lab and people are talking about some of these procedures, you have somebody in our class right now that's doing some fish. See, this is fluorescent in situ hybridization. And it would be nice if you actually heard the name of some of these things so that you don't have that look. So we'll definitely do this. Oh, I wish I had one of those because it's close to one of those. And this, I think we can get this done pretty quick. That's fun stuff. Okay. I think we'll call it for now. What's our time at? 5 viewers, thanks. Hopefully other people watch later on. I can't tell what my time is. It was 20, 30 minutes, been half an hour. I don't want to start the other one in person, so I'm just going to do it. I will see you on Wednesday. Have fun.